Hello, everyone. Welcome to our lecture number 7 on single-cell high-throughput data science. So today we are going to talk about single-cell RNA-seq differential expression analysis, so-called DE analysis, DE. So this has been a well-studied analysis, DE analysis in bulk RNA-seq, and this is a pipeline. So I will quickly walk you through this pipeline that has been used in many bulk RNA-seq analysis. So if you have a raw sequence data, FASTQ files, and pass through the QC, and then you do this short range mapping, and you map them using either splicing aware mapping approach, like bowtie or BWA, unspliced mapping or spliced mapping, use TopHat and bowtie 2, and then you can get the BIM file, and that's the mapped arrays in a BIM file. And then you can do this expression quantification. Basically, you count how many arrays was mapped in each different genes. And then you normalize it, use either FPKM or RPKM. Then once you have this array count matrix from more than one samples, then you can conduct differential expression tests using some R package or CuffDiffs. This is another R package. Through the comparison, you can identify the genes with expression difference between two samples, and then you can do the downstream functional interpretation, see what set of the genes have been upregulated or downregulated. You can use functional enrichment test, or you can infer network about the gene regulation co-expression, or you can integrate with other data to learn the biological insights, and then have a new hypothesis in which gene set you can discover, and then to design new experiment to validate. So the key output of the analysis is a list of gene called the DE gene. The gene changed its expression between the two conditions, two samples to compare. There are many methods for traditional RNA-seq DE analysis. Most of them are based on the count, because the DE analysis is based on RNA-seq data itself is count-based. So those are the established methods and been published and been used widely in the field of the traditional RNA-seq analysis. Some of these packages are still used in single-cell RNA-seq as well. However, there are different properties, such as whether the expression level contains a lot of zeros or not, because single-cell RNA-seq data contains a lot of zeros. It's a sparse matrix compared to traditional count-based RNA-seq matrix. So there are many aspects of difference in the design of the package. And there's one thing in common that people, statisticians, has been studying this, how to model the count-based expression level. As we mentioned, there generative model mathematicians, statisticians want to use to simulate or to approximate the real data. And they found this negative binomial among many other distributions is mostly closely related to this count-based expression matrix can be used to describe distribution of the observed from there. One thing is a discrete instead of a continuous variable probability distribution. So this is directly applied to those count data. And this map is a relationship between them, between different probability distribution. Okay, as we mentioned before, the negative binomial distribution described a process that that will be a good fit for RNA-seq data, both bulk RNA-seq and single-cell RNA-seq. That's what a statistician has been found, and there's a consensus among the field. So in negative binomial distribution, there are some parameters. K is the number of success, and R is the number of failures. So before you get numbers of K success, and the P is the probability of success. So we have used this as an example in previous slide lectures to see, okay, this is a number, the probability of detection, and that's number of undetected expression. Then you have a detected expression of the gene. So these three parameters fit this negative binomial distribution probability function. So we can use a summary statistics as a mean and variance to fit the distribution of your observed counts. So there's two variable mean and variance defined by the basic parameters of this RP. And then the combination of these two and give you the distribution of your probability of your data observed. So you can use your observed data to estimate these two variables and to fit your data to model the likelihood of the observation in your real data. So this is some plot when you have a established R. So R is fixed. Once you fix R, once you have a different P, and then you get a different shape of this distribution. So you got to find one of these combinations to give the best fit for each cells, genes, each gene's distribution among cells. Okay. So in DE-seq, which is one of the R package people use a lot for traditional RNA-seq DE analysis. So this estimates the mu and the variance sigma squared, and then fit this distribution in order to take into account both the technical variability and biological variability. It rebuild this shape of the boundary to everything within this area is expected, basically. So you consider once you have a high expression of gene, so something like this 10,000 compared to 100, and compared to even lower number, the variability is small when you have a high expression. And then, so if you have a gene which jump out of this boundary, a gene like this, have a higher fold change than expected, then this is a significant DE gene. But when you have a low expression on average for this type of gene, for example, this one, it requires the fold change. So be much larger than the gene with high expression on average in order to be reported as a significant DE gene, because we consider the wider variability among this lowly expressed gene. Okay, so this is a pipeline of the single-cell RNA-seq analysis. We have already showed this a couple of times, and we start from the generate raw data and you process this count matrix through the QC normalization and optional feature selection. You reduce the dimension and visualize it. And then you can do the clustering analysis to identify the clusters of the gene cells. And then you can use marker gene identification to find the cell types. And then once you decided to compare the gene expression difference between different clusters, then you can use DE expression, differential expression analysis. So in the next lecture, we are going to talk about this trajectory time series analysis. You can also have this composition analysis. That means if you have two samples, you will see the change of the proportion of the different cell types across two different samples. So in this case, the DE analysis can be applied to the same sample but a different cluster. You can also apply DE analysis for the same cluster across different samples. So there, for single-cell RNA-seq analysis, you have two different ways to use DE analysis. Compare cross cluster for the same sample. So for the same sample, you want to compare the green cluster against the red. So you can use a DE. You can also compare the difference between the same cluster across different samples. If you have multiple samples, you can do multiple way comparison, use DE analysis. Okay. Let's look at the DE analysis applied to the same sample across different cluster. You probably will get a heat map like this. Okay, this is from a single sample. Make it clear, single sample DE analysis. So you have a large cluster as a cluster 0 compared to cluster 1. So those are the cells. You have more cells in cluster 0. You have relatively fewer cells in cluster 1. And your comparison may help you to identify a group of genes that here. So those genes, for example, are relatively highly expressed in cluster 0, but lowly expressed in cluster 1 or other clusters. And then look at the next block of genes, those genes highly expressed in cluster 1 and relatively lowly expressed in cluster 0 or cluster 2, 3, and those are the results from a DE analysis. Then similarly, you can identify a cluster, a group of genes highly expressed in cluster 2 and relatively lowly expressed in other clusters, cell clusters. So this is the application of DE analysis to identify differential expressed gene in a single sample. So you can have result summarized using this heat map. With those information, you can assign cell types, and because you have a non-marker gene database, and you can assign the cells based on their expression of gene and the marker gene expression using either single R and the cell assigned to other R packages can apply and use them to assign cell types. As we mentioned, if you have two samples, say you have sample 1, and then you have several clusters, and you have sample 2, you also have some clusters. And so if you know this is cluster 1, 2, 3, and similarly this 1, 2, 3, and if you compare and you realize, okay, cluster 2, the number of cells was, see, 20% in sample 1, and it becomes 60% in sample 2. So this is called composition change. So you can find the differential abundancy of the cell types across different samples. And for example, this cell type 3 in cluster 3 become smaller in sample 2 compared to sample 1, and cluster 2 become more abundant in sample 2 compared to sample 1. So this is a so-called differential abundancy analysis. It's probably the first thing you want to do if you can somehow reliable collect the representation of the cell type abundancy. Then the method, which in HR, for example, can help you to decide whether this change is significant or not. So this probably is something even beyond the differential expression. And then you want to first count the proportion change, composition change across two different samples. So after that, if you find there's no independently, if either you found the composition change or not, you can next step you probably want to use differential expression analysis applied to single cell RNA-seq. If you have data from two different samples. So the purpose of this is to identify the genes that are differentially expressed between conditions within cells or from the same cluster. Remember, this from the same cluster means they are probably the same cell type. You don't want to compare apple to pear. You want to compare apple to apple, not orange to orange, instead of compare between two different cell types across two different samples or conditions. So that's the purpose of the DE analysis for single cell RNA-seq. You can use DE method designed for the bulk RNA-seq. Sometimes it's fun, and but we want to emphasize their DE method specifically designed for single cell RNA-seq. The mostly widely used is called MAST. MAST has been published in its R package. There are many others, and we will mention it, but MAST will be okay for most of the user cases. Sometimes you still need biological replicates to do DE. But this is not a so stringent requirement for single-cell RNA-seq. Number one is the cost. It's so expensive, so you may not be able to afford to do the multiple biological replicates of a single-cell RNA-seq sample. But on the other hand, if you don't have the biological replicates, it still be doable because you have so many cells have been sequenced from even one samples. Okay, there a problem before you can run the analysis package. This problem is called batch effects. So this is assumedly is generated from a non-biological factors and bias the distribution of this cell representation on this tSNE maps. For example, you have a control sample and you have treatment sample. They are clearly separated into two large patch of this distribution on the tSNE. If you don't do the correction, you may think, okay, the majority of the genes in control group will be differentially expressed compared to these treatment samples. However, this difference, global difference, may caused by the non-biological reasons, such as your prepared libraries slightly different and you did the experiment on the different days. You may use slightly different reagents and even the same set of reagents. You may have still have this technique batch effects. So you want to correct for this artificial separation of the cells and make them globally indistinguishable. So if you can correct the batch effect, you will end up with this so-called aligned database datasets. You see that globally there's no difference between these two sets of the cells collected from two different size batch. They are the distribution of these two data clouds are superimposed onto each other. There's no batch effects anymore. So once you achieved this step, you can start to use correct the batch effect removed data to compare these two conditions that identified genes that really express differently, not globally different. So the further description of the batch effects. So in molecular biology, batch effects occur when non-biological factors in experiment cause changes in data produced by experiment. So effects can lead to inaccurate conclusion when they are correlated with one or more outcomes of interest in the experiment. So this effects from a technical variation will confound the biological signal in single-cell RNA-seq analysis. So this is the same problem in the bulk RNA-seq as well. So in traditional RNA-seq, if you want to correct the batch effects, people use a linear regression approach that's implemented by in a package called Combat. So with single-cell RNA-seq, people also developed other methods to algorithm to correct for the batch effects. So we are going to mention two of them. One is an MNN and another is a CCA. So this slide describes the traditional batch effects correction for bulk RNA-seq analysis, because people still try to use the combat and SVA method for single-cell RNA-seq. And this tool is batch effects correction method developed specifically for single-cell RNA-seq datasets. As I mentioned, one is called MNN, is mutual nearest neighbor, and published in this paper, and also another is canonical correlation analysis, CCA, published in the same journal, same time. So I will briefly mention what is mutual nearest neighbor algorithm for batch effects correction. As I mentioned, it's published recently in Nature Biotechnology. It's based on the idea of KNN. We are going to describe KNN it's a very easy, simple concept. It's based on the distance between data points, and you found number, the k defined by k numbers of nearest neighbor, and make connection between these data points. So for instance, you have your first batch of data in the 3 clusters like this, and then you have your second sample, which collect information from another batch. So you have 2 different batches of data. So once you reduce the dimension and try to group them together, you found they are separated into 2 different batches. In what you can do, the first is to find each of these cells, data points in batch 1, and found the nearest neighbor of the cells, data points in batch 2. So if they are the nearest neighbor is this, you establish the relationship between them. And then the other way around, you start from the batch 2, and then you found nearest neighbor in batch 1. So you do this 2 ways search for the nearest neighbor using the distance measure between data points. And at the end, if these 2 cells, one is from batch 1, another from batch 2, they both found each other as the nearest neighbor during the search, and this is called mutual nearest neighbor relationship. Then you can establish a link between these 2. On the other hand, if you found this nearest neighbor from batch 1 to batch 2, but not the other way around, then this is not a mutual nearest neighbor. So you will not make a link between these 2 cells or these 2 data points. So once you established those mutual nearest neighbor relationship, you can link those cells from 2 different batch and make them as a nearest neighbor for each other. Then you do a correction between the linked cells. And for example, you add or multiplicate a fraction, a constant, to make them combine them into 1 cluster. So after that, you correct for these 2 clusters and leave some others uncorrected. And that's fine, because if you have another batch of data, you may have found the mutual nearest neighbors between another batch to this corrected which linked from another set of clusters. So in this way, you found the nearest neighbor across batches and then correct for their gene expression and eventually merge them if they are separated by the batch effects. And you merge them correctly, not introduce some artificial correction, overcorrect those, because if those cluster has no mutual nearest neighbors, they will not be correct for. Okay, so another technology people use is called the CCA, canonical correlation analysis. So you have two groups of the treatment, and then generate two separated samples, and when you combine them, you found they are strong batch effects in your data, and the red and the green cells are separated into two different locations in the tSNE. And then the CCA will help you to merge them and align them onto each other. And you can then use so-called dynamic time wrapping, DTW, to correct for their relationship, and eventually you get this so-called aligned data sets, and then you correct for these batch effects. So it's worth mentioning what the CCA is. So CCA is a method to correlating linear relationship between two multidimensional variables. So you have two multidimensional data points, that's single cell RNA-seq expression data matrix from multiple cells. So you can use CCA to do this feature selection. Basically, you want to extract information and to match them and to make so-called two views of the same semantic object to extract representation of semantics. Let's explain what's the views and what's the semantic objects. So you may have a data point from a completely different views, and in this case, one from this red, another from a blue, and then you can merge them in a common feature space and based on their semantic relationship. So that's the purpose or the goal of the CCA analysis, to find the trajectory direction that try to maximize the correlation between two views projected from the same instance. So two views, you can see, okay, one is the image, for example, and another is their the voice signal. You want to match them. Can be pictures and the description of those pictures. So there are totally different views of the signals. One is picture image, and then another is description using text. And another example is a view from the EEG, and then the view from a fMRI. They're completely different signals, but on the same objects. But you can probably find the link association between this data collected from different views. So that's the idea of CCA, to find the shared semantic space to maximize their correlation. So how exactly this can be done is, once you have this representation of two views of data, in our case is single-cell RNA-seq distribution data from sample 1, I can sample 2, then use CCA to find the projection to maximize the correlation between them. So you will need to compute a correlation matrix out of this transformed data points in the new space. So that's Matlab function. It can use very easily, and you put your matrix X, matrix Y. You found this A and B matrix to help you to rotate each individual input matrix. The correlation between the relationship between CCA and PCA and the other two technology, one is partial least square and the multivariate linear regression. Is the difference between this object A and B matrix. So CCA, PCA can be regarded as a special case, which there's covariance matrix and identity matrix for A and B. CCA use these two, and they all rely on SVD, generalized SVD eigen component operation, and to get the answer. And then, as I mentioned, after CCA, you can use dynamic time wrapping, and this is a technology used in the time series analysis to calculate optimized match between two given sequences. Okay, use the time series data analysis. One example is this: you have one time series data like this from time 0 to 1000. The one curve is this blue curve, and then another is this red curve, orange. And then after this this wrapping, and then the distribution of this red curve has been matched to this blue curve, because they do have similar signal patterns, and you can use this DTW to wrap them. So exactly how this DTW algorithm works, you can follow. There's a YouTube tutorial explain this details. Okay, as I mentioned, this when you have an uncorrect data, then you have different batches, and to show their slightly different arrangement in the two-dimensional space here, and the combat can do a job, and MNN or CCA, which is a method implemented in SURate package. SURate package is the most commonly used, most popular used R package to do this single-cell RNA-seq data analysis comprehensive package. They can do the job and produce slightly different results, but normally they are okay. So this is the first step you want to consider to use if you detect a strong batch effect between your samples, two samples, before you can start to use the analysis. Otherwise, the differential expression analysis will produce a lot of false positive results. They all come from batch effects, which you don't want to be confounded, to be included, to confound your real signal from biology. So there are different tools people can use for the analysis for single-cell RNA-seq, as we mentioned last, and published in 2015, is based on the generalized linear herd model. And it's R package, so that's the selection, and I suggest you will use. You can also have options, so-called CDE and Monaco. So in the early version of Monaco, they have a DE analysis capacity. There are several others. These two is from the traditional RNA-seq. Okay, so I mentioned last is the option of the DE analysis you probably want to use first to do your analysis. So it's based on the so-called generalized linear hurdle model to account simultaneously for stochastic dropout and characteristic bimodal expression distribution, okay, in which expression is either strongly non-zeros or non-detectable. Okay, the first we need to know for this model is I is cell, so I for cell I is individual cells, and this matrix X contains a matrix called the cellular detection rate. Okay, this contains information in how many rates tend to be generated for each cells. So this is statistics that link to the both of the detection power and also used inside this description of normal distribution for whether this can be detected randomly by this process. Okay. So the first, it used a logistic regression for the probability of whether this is a non-zero count, or for one gene in the cell I is a non-zero count or not. So this can modeled by logistic regression. So once you have this, you know it's a non-zero count. Then you have a probability whether you will get this number of count in your observation. So that's the formulas for the model, and then we will look at what is in the CDR, the cellular detection rate. Cellular detection rate is this Z for this cellular detection rate is for each cell. So for each cell I, there's a cellular detection rate. It's the summary of all the non-zero expression across all genes from gene 1 to n, then times n, okay, and then 1 over it. So this is a rate for detection. In the statistics field, people also use so-called zero inflated models. So they are slightly different from the hurdle model used in math and this zero inflated model. So the zero inflated model combined with the Poisson distribution is also another complex distribution people use in single-cell RNA-seq modeling. So there are subtle difference between them. This forum thread explain the difference. Okay, so next tool is called scDE, which you can find from the source code and how to use it. So I'm not going to details about this tool. Is based on the inflated negative binomial process. Okay. One thing I want to emphasize is for single-cell RNA-seq, we not only look at the DE, we should not only look at the DE. And in the DE, it's easy. So in one condition, this gene's expression increase or decrease, there's a very clear separation. However, you can have a different multimodal distribution. So in condition 1, you may have a distribution, density distribution like this. Okay, in condition 2, the peak changed, but it still has bimodal. And you can also have this differential modality. Then it was a single peak, however, it become a bimodal. There's 2 peaks, so this is a kind of change with both of the mean difference and also distribution shape change. And also there's a differential modality and the proportions. And then here the mean of these two will not be different if you only look at the mean, because they all centered in here. However, if you look at the distribution, one condition there's a single peak, another become 2 peak. There's no difference between them. It's only the proportion changed. So we have to take those into account. So there are many other tools that have been developed to consider those differences. One of the example is this called DScan. So this is published in the PNAS to do the deconvolution of single-cell expression distribution using a more sophisticated model, and the authors try to explain the process uses illustration that you have an original true expression distribution that all the cell size, the cell cycle has been in there. And then once you sequence it, you introduce both technical noise and batch effects. And then you have your observed UMI count matrix, and the distribution looks like this. And then this software tool, this algorithm, try to recover if you have some spike in, you add in some standard expression molecular to follow the distribution change. Then also they try to rebuild, do library size normalization. Then you try to rebuild this distribution and as to match the original true expression distribution. And they also take into account of the covariance. Okay, those covariance effects can be later to started, such as the size of the cell. And once you have this clear and feasible description mathematically and statistically about those distribution, then you can start to learn whether there's a distribution or characteristic change in bursting frequency, for example, in the dispersion level, and the differential tests. You can based on both of this frequency change of the non-zeros and then the change of distribution, dispersion difference. Oh, this is the most sophisticated tools we have. Okay, those two links to give you some extended information about RNA-seq analysis procedures. For example, in one of these tutorials, there are the analysis in a real data set. Then you have introduction of this. They load the real data in, and then the first test they use is called the KS test. It's a standard test for the two different distributions. So whether there are significant different distribution between their CDF plots. Then they use this Mann-Whitney U test, which is a standard test for the non-parametric distributions. See whether there are difference in the count data. You can use this as a quick approximate for the difference between two conditions. And then they show how to use HR, which is a conventional differential expression data applied for both bulk RNA-seq and single-cell RNA-seq data. And then they show the monocle and have a several function for differential expression analysis. Then they should use MAST. So that's some code you probably want to use. And then there are other examples. So this is just one of the tutorial people put them together and to teach you how to use these tools. So that will be a good reference for our applications. And that's all for today's lecture. And see you next